Hello students. Previous session we talked about what are the different digestive glands and what are the parts of the digestive system and why these digestive glands are important. Now in this session we are going to look at how the process of digestion takes place and what are the different enzymes which help in the process of digestion. To look at these subtopics, we will be taking down the subtopics. The first one will be what are the digestive enzymes which help in human digestion. So different enzymes from different areas we are going to talk about. Next one will be what is the first part of digestive system where the digestion starts, that is at the mouth region. Now the third one will be why is villi important for the process of digestion, and the fourth one will be what exactly happens with the large intestine. Now these all will be covered. Before we move forward into the process of digestion, let's look at the various enzymes and important secretions which happen in different parts of the digestive system. Now the first one here on the screen is you have already seen this, they are the different parts of the digestive system. To start with, we will start with the mouth region where the salivary glands are present which pour out their saliva, and the saliva contains an enzyme called salivary amylase. We'll start with this. First looking at the buccal cavity. So buccal cavity in saliva, we have the salivary amylase. And another name given to this is the ptyalin. So this will be the enzyme which acts upon carbohydrates, that is the complex carbohydrates, where they will be broken down in between substances which we call them as double sugars. Now here in the liver region, liver is also a very important gland, but it does not have any enzymes as such. So what it has is only the bile secretions. So we call it as the bile juice, and bile juice is important as such because it has a major function to handle with the emulsification or the breakdown of fats. Now the third region will be the stomach region. Now in stomach there are three different things which are being produced. One is the HCl, the other one is the mucus, and the third will be the pepsin enzyme. Now this pepsin acts upon proteins. It acts upon proteins. Now coming to the pancreatic region secretions will give out the final products of digestion. So we have three enzymes we are looking at here: the trypsin, the amylase, as well as the lipase. That will be from the pancreas. The bile juices along with the enzymes of the pancreatic juices will be poured at the junction of the duodenum, which is the starting point of the small intestine. Now that will be the area where the initiation of these enzymes will take place. Now coming to the large intestine does not have any enzymes or any digestion to be done because the digestion completes by the end the small intestine ends. Now large intestine only has to reabsorb the amount of water. So the water reabsorption takes place at this particular area. The undigested matter, what we call as the feces, will be continuing forward to the rectum, and rectum it will be stored temporarily before the expulsion happens through the anus. At every point in the digestive system, let it be between the esophagus and the stomach to the small intestine, everywhere you find there are constantly valves which are very important so that they close down once the digestive products move ahead. Now the sphincters are also present between the small intestine and the large intestine at the region of the appendix here, what we have given. So there is a sphincter also here, and then between rectum and anus, and in anus there are two sphincters to guard that opening. Now these sphincters are very important because we do not want the digested matter to get back in the same process. So with that understanding, let's look at the different enzymes and the action, what they are doing to complete this process of digestion. Look at the digestive enzymes. So we already studied that the digestive enzymes present in different areas. So we will be starting with the salivary glands. So salivary glands, we said it produces an enzyme called salivary amylase. Now this salivary amylase has a neutral to an alkaline kind of medium, so we cannot handle acids in the mouth region. So salivary amylase will convert the starch, that is the complex carbohydrate, into double sugars like maltose. So the process of digestion is only partially completed at this particular junction. So maltose is a double sugar. So coming to the next one, we will talk about is the stomach region. Now stomach, we said there are three things, that is HCl, the mucus and the pepsin. HCl creates an acidic medium because in the mouth we said it's neutral to alkaline. Now this acidic medium is very important for the food which comes in the form of bolus, which is a crushed food pushed through the esophagus to the stomach. Now this HCl gives an acidic medium to the food. And then when the food enters, there will be a lot of germs also present. To kill these germs, this acidic medium is important. And the most important one of HCl is the making of the pepsin enzyme in its active form. We all have enzymes in an inactive method or inactive form. To actually make them active, they have to be changed into that particular form. Now this another function of HCl is activating the pepsin. Already highly acidic, we do not want to make it more acidic. The more acidity happens in the stomach region, the more chances of rupturing the mucus layer of that stomach region. So which causes bleeding ulcers, where the stomach region start bleeding at that position. We don't want to make it more acids by adding more unwanted foods. Now coming to the mucus region, mucus is very, very important in the digestive system because many of the areas are protected. Now stomach region also is similar. So the HCl which acts on the stomach constantly will be touching the walls of the stomach region. So to protect this layer, we need this particular mucus. So mucus protects the walls of the stomach. Coming to the next one, the pepsin. Pepsin is the enzyme which comes in inactive form, converted by HCl into its active form. Pepsin acts upon proteins. Now this is the area where first the proteins will be acted upon. They are converted into partially digested products, which are called as the proteases and peptones. So now we have seen that the carbohydrates are partially digested to maltose and the proteins have been converted to proteases and peptones. This will be the initial first step of digestion of these two substances. Now coming to the next one, the bile juices. Bile juices, even though it does not have enzymes, it has two pigments which are very important, that is biliverdin and bilirubin. Now these two pigments add coloration to the feces. Now the next function of bile juice will be the emulsification of fats. Fats have to be broken down into smaller substances so that the enzymes of the small intestine have to be broken down. So into the final products of digestion. Now this will be the function of bile. Apart from bile, the pancreatic juice and the bile juices will make the food into alkaline medium. So alkaline medium, the change of acidic to alkaline from the stomach to the small intestine also happens with the bile juices and the pancreatic juices. Now coming to the small intestine. Small intestine will be the area where the final products of digestion are being released. For that, we have to understand what happens in the small intestine and what are the finger-like projections which are present in the intestine, which is useful for digestion. Now, Now these are the villi of the intestine. So these are the parts where the foods which are digested completely in the small intestine are absorbed. Now these are finger-like projections, what you see on the screens, and you also have lacteals which are important, which help in the digestion of fats. So we said the bile juices will break down the fats into smaller substances. Now those substances will again be broken down into simpler products before the lacteals come into the rescue to absorb those simpler products. Now let's look at what happens in the small intestine and what are the different enzymes which act upon here. The first one will be the trypsin. Now this trypsin, whenever we have trypsin, erepsin, when it ending, it always acts upon proteins. Now this trypsin will break down already digested that peptones, proteases in the stomach, and along with the leftover proteins, it is finally digested into amino acids. Now the final product of digestion for proteins will be the amino acids. That is the simplest form of proteins. Now the next enzyme we look at is the pancreatic amylase. Now this pancreatic amylase will digest already done double sugars, that is the maltose and any kind of double sugars. All that along with leftover carbohydrates or starch material, the final product of digestion will be simple sugars. So we can say the simple sugar to be glucose or any simple sugar. So the final product of digestion of carbohydrates from the complex substances will be the simple sugar. Now this will be the second product which is being released at the small intestine. Now we have said lacteals here. So there is an enzyme for the digestion of fats which is called lipase. Now what does this lipase do? It will be already broken down by the bile juices. It will be breaking down into further simple substances. So the fats which are already smaller are broken down into fatty acids and glycerol. Now this fatty acids and glycerol will be the third product of digestion at this small intestine. Now but this fatty acids and glycerol will directly be taken up by the lacteals which are present in the villi. So villi major functions, it acts as a medium between the tissues. Once the food is digested, it will move it to the tissue so that it can be circulated through the blood region. Now here, apart from this, it also increases the area of absorption because that is the place where the complete digestion takes place. Now these two will be the function of villi, and you can see there are a lot of capillaries and arteries also in the diagram. They all help the particular villi to be always having this exchange of substances. What happens in the final products of digestion? So after this, we will have only the large intestine. So we already studied that the large intestine only has to reabsorb the water if at all the feces are not in the correct form which has to be expelled. So reabsorption of water takes place, storage takes place in the rectum, and final expulsion through the anus. Now coming to few questions we will be taking up. Now what will happen if mucus is not secreted by the gastric glands? So we have already understood if mucus layer is not present, the walls of the stomach have high chances of getting ruptured, and then they might turn into ulcers called as the bleeding ulcers. Now that will be the first answer. Now where does the final digestion takes place? We have already studied the final digestion takes place in the small intestine. So you can write the final products of digestion, the amino acids, simple sugars, and fatty acids and glycerol. So here you can take, it might cause ulcers or the rupture of the stomach region. It has to be protected. Now where does the reabsorption of water take place in digestion? Now this was the question. Now reabsorption of water finally takes place in the large intestine. So these are the three questions. We will look forward to some more questions. Which organ helps in the emulsification of fats? Fats are all broken down by the bile juices which are being released from the liver. So the bile juice will help in the emulsification of fats which is released by the liver so that they can be broken down into simpler substances so that the lipase will work into much simpler products. The next question. What are villi and how they are related to the digestive system? Villi are the finger-like projections. We already seen finger-like projections which help in absorption of the final products of digestion. Which help in the absorption of final products of digestion. Now coming to the third question, what are the final products of digestion? The final products of digestion we have already seen that the trypsin, all of them, that is the trypsin, amylase as well as the lipases will break them down into simpler substances, the amino acids, simple sugars as well as the fatty acids and glycerol. So to conclude this session, we have seen what are the different enzymes which help in digestion and how they are made into simple products because it has to be absorbed through the body and circulated through the tissues present in the body. Now we will be looking at in the next session, we will continue this life process in the next session, and for now, thank you.